Okay, so today we'll look at this part. Okay, so yesterday we have talked about the process of DNA replication. Okay, so just a very quick recap. Okay, so a few things to know about DNA replication. Okay, so of course before DNA replication, the nucleotide need to be prepared in the sense that they need to be activated. So that activation occurs through a phosphorylation of an nucleotide. Okay, so adding another additional phosphate group to this nucleotide. Okay, of course, this is going to help it for incorporation into DNA to be synthesized. Okay, and for DNA replication to occur, okay, so I said, okay, so it begins at what we call the origin of replication. Okay, so the DNA polymerase binding to to some parts of the DNA, okay, of the chromosome to initiate, or chromatin to initiate DNA replication. Okay, so under this process, okay, so there is this enzyme called helicase, which function to just, basically it functions to separate the two strands of DNA so that they can be read. Okay, so in this process, hydrogen bonds are broken. Okay, so your parental strands are unwound. Okay, so then for the synthesis of DNA, okay, just one thing to note, okay, as we have highlighted yesterday, that your two DNA molecules, firstly, they are anti-parallel, one running from 3' to 5' direction, and the other one, the opposite one, in an opposite direction. Okay. Secondly, these DNA polymerase are configured, okay, or as in like they have evolved to only read your DNA in 3' to 5' direction, while also synthesizing DNA in the 5' to 3' direction. Okay, so for this reason, okay, it presents a bit of a challenge, as in like a little issue, okay, for DNA replication. Okay, like I mentioned yesterday, okay, so when the DNA is unwound, okay, so this is what we call a replication fork. Okay, so you have two DNA polymerase binding to two different strands. Okay, but because of the anti-parallel nature of your DNA, okay, so of course for one strand, it will just read along, keep going in this direction, in the 3' to 5' direction. Okay, 3' to 5' direction, while also synthesizing from 5' to 3'. But the other one, it would go in an opposite direction, which means in this case, even though this part is open up, at least for one of the strands, this region wouldn't be read. Okay, because this DNA polymerase will go in the 3' to 5' direction rather than 5' to 3'. Okay, so therefore, when the DNA replication fork, okay, open further, okay, then it allows binding of another DNA polymerase, which will then read again in 3' to 5' direction. And in this case, at least for one of the strand, okay, the synthesis of DNA occurs in fragments, because here it starts and then synthesize here, and then another DNA polymerase is going to bind later and synthesize another fragment. But these two fragments are not joined together. So these fragments are called, what are the names? Okazaki fragments. Okay, it's discovered by Japanese. Okay, so of course these fragments ultimately will need to be joined together by an enzyme called DNA ligase. Okay, so it's like a molecular glue, just glue things all together. Okay, so that's kind of the gist of DNA replication. Okay, so, and then of course at the end of the replication, okay, so the newly synthesized DNA is going to, they are going to coil together to form this double helical structure. Okay, and this whole process, as we discussed, is really semi-conservative, which means half of the original strand will be preserved after the next round of DNA replication. Okay, so now this is Michelson and Stahl experiment. Okay, so this experiment provides evidence for semi-conservative replication model. Okay, so what they did was that, okay, so just FYI, there are ways to chemically synthesize certain compound, okay, with the certain isotopes. Okay, so here the medium in which the bacteria are grown, okay, the media is supplied with for example, with nucleotide, okay, which is synthesized using the isotope 15 of nitrogen, okay, and 15 isotope, which is a heavier version of nitrogen. Typically, nitrogen at relative atomic mass is 14, okay. So you would grow, so what they did was that they grew these bacteria in the N15 medium for a very long time, which means, okay, over time, you probably expect all the DNA within these, all the DNA within the bacteria heavy end, okay, comprises the heavy end isotope, okay. So after that, what they did, they will just transfer this bacteria into a different medium with the light nitrogen. Okay, so let's see what's going to happen here. Okay, so of course, at the end of the experiment, okay, so in a different, maybe for each round of replication, whenever they see, for example, the DNA, the amount of bacteria double, then they just harvest them. Okay, so every time when they double in number, they harvest and then extract the DNA. Okay, what they did then was to add them into this particular solution called cesium chloride. That doesn't matter. Okay, and then centrifuge them. Okay, centrifuge is centrifuge, you know, like centrifuge. So centrifuge is a machine that basically just spins things very quickly. Okay, so in physics you probably have come across a centripetal force, centrifugal force. Okay, so basically it is just spinning things. Okay, of course, whatever that's heavier will sink to the bottom. Okay, so here, let's see. Okay, so let's say at the first generation, okay, all of those, okay, when they were first, okay, before they were transferred to the light medium, okay, some of the bacteria are taken out, they are broken down, okay. So then you spin the DNA, okay. So because all of these are the heavy version of DNA, they would go to the very, very bottom of your of the test tube, okay. So then after that, they transfer them to the light medium, grow them for one generation, as in like allow them to double in number. So there's one round of DNA replication, and then spin again. Now they realize, oh, the DNA become lighter. Because why? Because of course, based on what we already know, of course, at that time they do not know it for sure. Okay, just imagine at that time, these scientists do not know for sure as to how DNA replication occur. Okay, but by simply extracting DNA, spinning them, okay, seeing the change in mass, okay, that gives some indication. Oh, so firstly they notice, so after the second generation, after one round of DNA replication, oh, the DNA that they extract become lighter than the parental generation, which is kind of expected in the first place because you have transferred them to light medium. Of course, it's going to become lighter, but the question really is, what is the percentage of light and heavy? Okay, so next they do the third round of them. Okay, so actually at this stage, they will find that 100% of the DNA are lighter. Okay, so and then another round. Okay, so don't look at the diagram first. Okay, I'll just say what happened when they did this experiment. Okay, first round, all heavy, sink to the bottom. Okay, second round, okay, as in like after one round of replication, DNA become lighter. Okay, so there's only one fraction. Okay, so all of them at the middle. And after another round of DNA replication, they realize, oh, now they've split into two portions. Okay, one is even lighter than the previous one. Okay, so then of course they perform some analysis of the DNA case, so, and then they can identify what is the percentage of the heavy and the percentage of the light one. Okay, so some of this, can you please tell me, based on the physics knowledge, how can this how can that be analyzed? Like, how do you know that what is the percentage of the heavy, or what is the percentage of the light? Any idea? At least for, but of course, perhaps not in this case, but just FYI, in physics, you probably learned before, okay, some isotope can be radioactive. Okay, so by measuring radioactivity, sometimes it can give an indication of percentage, but I don't think N15 I don't think N15 is actually how should I say, is actually radioactive. Okay, anyway, so they have some way to analyze. Okay, so and one interesting thing is that you will find that the half, so things always decrease by half, 50%, 50%, 50%. Okay, so I'm going to delve into data later. Okay, so we see always there's like a 50% change. So 50% is like so that's something very consistent, because if let's say, because there are many ways people imagine how DNA replication can occur. Okay, maybe you just synthesize a whole new one, okay, without conserving anything from the past. Okay, theoretically speaking, you can use the 2 strand as template, okay, synthesize two new strands and keep the new strand together, which means, okay, you have a wholly new DNA, okay? But of course, in that case, do you think you would have this? Okay, so let's say, imagine, okay, so let's say this is your parental, okay, so let's say after replication, so let's say this is what you get, okay, if this is what you get, okay, if this is what you get, how many portions of DNA do you expect? So when you do the spinning, okay, so of course this is N15, this is N14. If you do the spinning, how many portions do you get? How many fractions of DNA? So would this and this, do they have the same weight, same mass? No, okay? So for sure, you may expect the N15 somewhere, okay? So you will still have the one at the bottom, okay? And then you will have one that is at the top, okay? This is the N14, okay? And then you have the N15, okay? If this is what happens, but instead, they got this. This is gone, okay? You lose the bottom fraction, and then everything become lighter, okay? So this is the first indication that, okay, the newly synthesized DNA is mixed, as in it's a mixture of heavy and light, rather than purely heavy or purely light, okay? So, of course, they do multiple rounds, okay? And then you can see how that changes, okay? So now let's have a look at this diagram, okay? So if you look at this, okay, so after the first round, okay, so these two parental is a red one, okay, so they separate, okay, as an old template. So semi-conservative, all the newly synthesized daughter will keep one of the heavy strand and then one of the light strand, okay? So 100% of them, this is a single DNA molecule. We call this a DNA molecule. So a double helix, okay, so we have 100% of the double helix are hybrid. Hybrid, which means half is heavy, half is light, okay? So if you, in terms of DNA content, 50% heavy, 50% light, okay? But in terms of the whole molecule, all of them are hybrid, okay? So that's why you get only one fraction of your DNA, okay? And then next, you do another round of replication, okay? So you will see when I draw this, if you look at this, okay, so you do see that, okay, because a red strand, okay, will be short, okay, will be diluted because as you grow them longer in a light medium, okay, of course, it's going to reduce in number. So you can see that, you know, so of course, here and here, so depending on, so this can become a template, this can become a template, right? Okay, so from here, you get a 100% O light, but this one, it will still be hybrid. Okay, same here, this one as a template, this one as a template. Here, 100% light. This one is a mix or hybrid. Okay, so for that reason, you have two fraction of DNA. Okay, in this case, after two replication, after two round of cell division or two round of DNA replication, now you get two fractions, okay? So there is a slightly heavier one, which is the hybrid, okay? And then you will have a totally light one, okay? So in terms of the whole DNA molecule, 50% because it is two hybrid, two O light, okay? So 50% hybrid, 50% O light, okay? But then if you look at the DNA portion, okay? So in terms of the fraction of DNA, if you don't consider whether it is, you know, in a DNA single, double strand or not, you see out of these 8 strands, 6 are light, 2 are heavy, okay? So 6 or, okay, so 2/8 and 6/8, okay? So that means, okay, you have 75% of DNA is light, 25% of DNA is heavy. So again, you see a halving, half, okay? Things are decreasing by half there, okay? So that provides evidence to support the theory of semi-conservative replication, okay? So that whole process there, okay, you can also, okay, I don't know if you've drawn this case. So yesterday case, so like I said, so this one grown in heavy, okay, so basically the same thing, okay? So and then after one generation, they are separated, okay? So and then each of these is going to serve as a template, okay? So then here I use green as heavy, red as light, okay? So you can flip to the next page, okay, for this one, okay? So just have this drawn. Can you please draw this? Yeah. And then you can answer the questions below. Or maybe, or you can just draw it from. I don't want to spend time for you to draw. Just attend the questions. Actually, have I not sent this to the group yesterday? Sorry? Oh, okay. I'll just send it again then. Okay, so you just copy that at your own time. Okay, so I mean for the drawing, you can do it at your own time. Okay, so I just want you to attend the questions below. Oh. Okay, so let's have a look. Okay, so here it says starting with 1515 heavy DNA, after 2 generations will contain, okay, so actually this diagram summarizes the whole thing. Okay, so after 1 generation, okay, so this is grown in 1 generation, you all of them 100% hybrid, 50% heavy, 50%. Okay, so after the 2nd generation, okay, after 2nd generation, this is what you get. Okay, so where you see that, okay, so we have 50% hybrid, 50% light. Okay, and in terms of the DNA content, 8 strands here, okay, like I mentioned just now, 75% light and then 25% heavy. So let's have a look at what's the question. Okay, so here, so 2nd generation, it is this. Okay, so 50% 15 14 and 50% 14 and 14 and okay, so basically whatever that summarized in the diagram above. Okay, so starting with this, okay, after 3 generations, okay, so will contain 75%. Okay, so again, this diagram show you that. Okay, so with this one, all of this, you have 8 strands here. So they'll give rise to 8 DNA double helix. Okay, so the red one will all be light. Okay, so those using the red as a template will all be light. The one using the green, which is a heavy, will be hybrid. Okay, so, and that will make up only 25%. So this, okay. So this one will be this, okay. So the 25% are the hybrid, okay. So the answer is 75%. Okay, so next we look at protein synthesis. Okay, so for this part, most of the stuff is something we have learned, okay? So we have already said that DNA encodes protein, okay? And of course, protein is made from amino acids undergoing condensation reaction. So yeah, and then of course, we have also learned that the structure, function, okay, your protein structure very much depends on the primary sequence of amino acids. So primary sequence will decide how they fold into the secondary structure and the tertiary structure. Okay, so which is why sequence of amino acids is very important, which also means DNA sequence is very important. Okay, So, and then a gene is what encodes for protein, okay? So your sequence of DNA will decide the sequence of amino acids, okay? Something we have already learned, so I'll just talk while you write, okay? And then the genetic code.
What sequence? Friends share coding. It's the same. They that.
Base sequence.
Okay.
Have you seen cursor writing before? Like normally,
Yeah,
like cursor writing S is right like this. Okay, so at the beginning of this chapter, when we talk about DNA, we've considered the coding, okay? So as we have learned, 3 bases would code for 1 amino acid, okay? So you have 3 nucleotide base defined, okay? So every 3 define 1 amino acid, we call it the triplet of the code, okay? So can you please tell me why it gives you 64 possible combination? This is your permutation, your maths. Okay, so 4 times 4 times 4, 4 power 3. Okay, because you have 3 position, each position has 4 possibility because here repeats is allowed, right? Okay, so there are 64 possible combinations, but you only have 20 amino acids, okay, 20 plus, which means you have extra code, extra capacity for coding. So therefore, later we are going to learn that, you will have multiple code for the same amino acid because like I say, you have 64 possible combination. You only have 20 amino acids, okay? So we have extra codes to use, okay? So later we'll look at that in detail later, okay? So the base sequence is always read in a 5' to 3' direction, okay? The base sequence refer to the sequence for the amino acids, okay? The one that code for amino acid sequence, because you have 2 strands, right? Only one of the strand would be carrying the gene information, okay? Not both strands, only one strand, okay? So here I just want you to know that, okay? So the 2 strand, one is called the sense strand, okay? So sense strand is the coding strand, which means it has the information for the protein sequence, for amino acid sequence, okay? And then the one that is complementary to it is called the antisense strand. And very importantly, I want you to know, okay? Antisense strand is a strand that is used for transcription. We'll come back to this later. What is transcription? Okay, but maybe I'll come back to this diagram later. Okay, so when we look at the subsequent process, we'll revisit this diagram. Okay, but have you taken this down?
No.
Okay, so but for now, I just want you to know that. Okay, so sense strand is a coding strand. Okay, sense strand is a coding strand. So next, okay, so each DNA base in a triple is only used once, non-overlapping. By non-overlapping or used once, what it means is this, okay? So let's say if I do AG's, okay, so if I have a sequence of A, T, G, C, G, T, T, okay, so whatever. Okay, so when we say non-overlapping, we read 3, 3, 3, okay? If it's overlapping, it means I will read AAT, ATG, TGC, GCG, okay? So that is what meant by overlapping, okay? So like this, if I read in these codes, so there are just 3 codes here. If you read in an overlapping manner, how many codes are there? if I read in an overlapping manner, how many codes are there? How many triplets are there? No, based on this.
3, 3, 3, 6, 8, 7.
7, 1, 2, 3, 4, 5, 6, 7. Okay. So if it's read in an overlapping manner, if it's non-overlapping, just 3 codes. Okay. So here just 3. So they are read in an non-overlapping manner. So that's what is meant by non-overlapping. Okay. So the same triplet codes is universal, which means whether you are talking about a plant or an animal or human or bacteria even, or even in, in the, even for viruses, okay? So it's the same code, which means, if AAA is phenyl, PHE is an amino acid, okay? If AAA is for phenylalanine, for all organisms, it's the same code, okay? So that kind of gives you an indication that this is something that is very, very, very old in revolution and has been kept for millions of years, okay? So some amino acids are coded by more than one triplet code, which is what we call degeneracy of code, okay? So like I mentioned just now, we only have 20 amino acids, okay? And then we have 64 codes. So that's why we have extra code to use, okay? We have extra codes to use. So all of these would code for, you don't have to write those in bring in papers, okay? So we have extra codes to use. So this is what we mean by degeneracy of code, okay? So you have multiple codes for the same amino acids, okay? So like alanine, you see there are 4 codons or 4 triplets for alanine, okay? So yeah, this is called threonine, also 4. So depend, okay? So yeah, each sequence of 3 bases, okay? So the sequence of 3 bases in DNA, we call them triplet of code. In mRNA, we call them codon. In tRNA, we call them anticodon, something that again has, which we have covered in the last 2 days. Okay, and also we have particular code for start and stop because the transcription or translation has to stop at somewhere. Okay, so translation has to stop somewhere. Okay, so we have some which we call stop codon, which means they don't code for any amino acid. Okay, we have some code which simply does not code for any amino acid. Okay, so if you see we have 3 stop codon. Okay, we have 3 stop codon. Okay, so they don't code for any amino acid. So when later we're going to learn when ribosomes come to this codon, no amino acid will be incorporated. Then the whole process of protein synthesis stop there. Okay, and start codon is always this. Okay, it is always a methionine. Okay. So note that many proteins, the first amino acid is always methionine. I say many because sometimes after synthesis, some amino acid is trimmed, okay, it's removed. Okay, we'll learn about that later. Okay, but otherwise, always protein synthesis start with the first one being a methionine. Okay, and that is okay, and then so okay. So this is on the mRNA. Okay, so above here, this is for DNA. So DNA, you have TAC. So if you do the translation, then it becomes AUG. T complementary to A complementary to U, C complementary to G. Okay, so on mRNA, it is always an AUG. Okay, on the DNA, okay, it is the okay. I want to say, okay, by the way, this strand, okay, this genetic code is on, I want you to also make a note, this is DNA and it is the antisense strand. Okay, the code that you are seeing here, please make a note, okay? So they are DNA on the antisense, like I say, there are 2 strands of DNA. One is antisense, one is sense, okay? Sense and antisense, okay? So what we have here are for the antisense, okay? So just make this note, okay? So, and also they are, we say degeneracy means redundant as in we have multiple code for the same, okay? Okay, so next, here, that's the central dogma. You can copy this first. Just 1 minute. Okay, so for there is this central dogma, okay, basically just like a main principle about this whole information flow process, okay. So basically it says, okay, so as we have kind of mentioned already, okay, so always you have information stored in the form of DNA, that's your genetic information, okay. So of course, DNA replication is a DNA-directed DNA synthesis. What it means is that whatever that is newly synthesized is based on the DNA sequence due to complementary base pairing. Okay, so your DNA replication. Okay, so it's DNA directed because this is due to okay, or maybe you can say made possible by complementary base pairing. Okay. Yeah, so your DNA strand will tell you what is the next strand, okay, what is the sequence of the new strand to be synthesized. Okay, so of course, as we know, DNA replication takes place in nucleus, but protein synthesis occurs in cytoplasm. Okay, so your DNA molecules cannot just go through because they're very large molecule. Okay, so therefore we have to find a way, copy that information out and then send it to the cytoplasm where protein synthesis occur. Okay, so for that reason, we only copy a little part of it, okay? So, and then bring that to cytoplasm for protein synthesis. Okay, so the copy form is mRNA. So mRNA synthesis is by transcription, okay? So, and then this mRNA is going to leave the nucleus, okay? And then they will be bound by the ribosomes where this protein synthesis is going to initiate. That's what I'm going to talk about later. So this whole process can go from DNA to RNA to protein. So that's how information flows. Okay, you go from DNA, copy to RNA, and then for protein. Okay, so this diagram also basically summarizes that. Okay, so always I like to give the analogy. Okay, so your DNA is like your CPU. Okay, all the information is stored there. Okay, so DNA is like your CPU, your central processing unit, and also where all your hard disk, all information is stored there. Your RNA is like your, like a pen drive, your USB disk, okay? So we don't need everything. We just need to copy a little piece of information out, okay? So you don't need to take everything out from your library or DNA, okay? So we just take a small piece of information out and use that to execute whatever that needs to be done, okay? So that's the kind of analogy I like to make, okay? So let's see, okay, so the first stage of your protein synthesis is that, or maybe before protein synthesis can happen, we have this transcription, okay? So DNA triplet codes are transcribed into mRNA, okay, using the antisense strand as a template. This is something very important, okay? So like we mentioned just now, let's say your DNA, you have 2 strands, okay? So this is a sense strand. So sense strand give you the sequence of DNA, sequence of amino acid, okay, so whatever. So maybe this is hold on. So this is maybe ATG or CGA or whatever. Okay, so this tells particular amino acids. Okay, so then you have the antisense strand. Okay, you have your antisense strand. This would have the TAC, and then this would be GCT. Okay, so this is antisense strand. Okay, so when performing transcription, okay, so when performing transcription, it is an antisense strand that is read, okay, so this antisense strand is read. So then when you synthesize your mRNA, this will be AUGCGA, okay, so this is your mRNA sequence. Okay, and what you will notice is that, okay, so this mRNA sequence will have the same sequence as the DNA sense strand, okay, but remember, when we synthesize mRNA, we use the antisense strand, okay, so that's why you see, okay, so again, one thing to know, your mRNA will have the same sequence as the same strand of DNA, except your T in the DNA is replaced by U in mRNA, okay. So basically, that's what happened. Okay, again, for this, for transcription to occur, that double helix need to be unwound or has to be separated. Okay, so hydrogen bonds are broken, then RNA polymerase bind. When you synthesize DNA, you use DNA polymerase. When you synthesize mRNA, you use RNA polymerase. Okay, so then RNA polymerase actually normally starts somewhere. Okay, so normally before your DNA, okay, so before your DNA sequence, okay, normally there's this region, okay, so there's some sequence of DNA, they are called a promoter region. Okay, so this is where RNA polymerase bind. RNA polymerase will bind here first, then they start to read whatever that's following it. Okay, so the promoter, just a promoter region, basically a sequence. Okay, so it's a sequence of DNA, or based on DNA. Okay, so just a stretch of them. So normally your RNA polymerase will only bind to certain sequences. Okay, and then they will then moves along the antisense strand in a 3' to 5' direction again. Okay, so it moves in a 3' to 5' direction, and then synthesize the mRNA in a 5' to 3'. mRNA will be synthesized in 5' to 3'. Reading will be in 3' to 5'. This is the same as DNA replication. Okay, DNA polymerase also moves along DNA in 3' to 5' direction and synthesize in the 5' to 3' direction. Yeah. Okay. And then so okay, so the free activated, okay, so what happened next is that, so this RNA polymerase will then move along antisense strand, okay, and then it will at the same time, whatever, so similar to your protein DNA replication, okay, so the mRNA will come in, okay, so depending on what sequence it is, okay, so let's say you have a DNA, so this is your antisense strand, you read along this direction, okay, so when you come to T, then an A RNA, adenine RNA nucleotide will come in. When it comes to an A and then RNA, they, not to say they come in consciously, like I said, all of this, okay, you need to remember, in cell, everything happens randomly. Everything actually occurs by chance, okay? But because A can form hydrogen bonding with T more easily, the rest cannot because of the complementary. So we talk about complementary base pairing. It is really down to shape. Okay, so I think at very early on I've emphasized to all the things. Okay, whatever interactions happening at the molecular level, okay, occurs through shape complementarity. Okay, which means your shape need to fit so things can join together or associate with each other. Okay, so for sure, okay, so maybe you have randomly A, U, G, C, whatever hitting coming into contact. Okay, but for sure A will form connection or hydrogen bonding with T, so they will stick around more easily, and then therefore they'll be incorporated into the strand of RNA that is being synthesized. Okay, so again, this nucleotide needs to be activated as well. Okay, so yeah, I think that's okay. So yeah, so when we say activation, okay, so just now we have mentioned, you add a phosphate group to the RNA or the DNA. Okay, so hydrogen bond then forms, okay, and then after that, so they will associate, and then RNA polymerase is going to, okay, so as long as there is hydrogen bonding formed here, so that RNA polymerase is going to form, is going to link them, glue them together, okay, so form by formation of phosphodiester bond, okay, so that is a phosphodiester bond, okay. And then, okay, so joined together by phosphodiester bond. Okay, so of course that catalysis of this, the formation of this bond is catalyzed by this enzyme. Okay, and then it will keep continuing until RNA polymerase come to what we call a terminator sequence, or simply by terminator sequence, it means the stop codon. So as we mentioned, stop codon does not correspond to any. Oh, sorry, ignore this. So anyway, so RNA polymerase will come to, okay, it will recognize certain sequence, which means when it comes to this sequence, it will just release it, okay? So, and release the mRNA. So, and then the DNA can rewind again, okay? So that's a copying process, okay? So here, part of the DNA is going to be unwound, for transcription to occur. So one RNA polymerase arrives at the terminator sequence, which means it knows, okay, this is where transcription should stop, then it will stop, okay? And then the mRNA will be released. And that mRNA is going to before that, okay, so one mRNA is transcribed, okay, so actually sometimes this mRNA may still need to be further modified, okay, so for example, mRNA has some regions called intron, okay, so actually the actual picture is more complicated, okay, so not the whole sequence will be used for protein sequence, for protein synthesis, okay, so there'll be some regions, okay, that will be removed. They are called introns. So all of this is RNA sequences. Okay. So some will ultimately lead to contribute to protein synthesis, but some of it will be removed. They are called the intron region. Okay. So then, so this intron will be removed. Okay, intron will be removed and then join the exon together. Okay. So then you will form. Okay. So introns are removed, and the process of joining things together, we call them we call that process annealing. Okay, we remove the intron and then join together the rest. So that is called annealing. Okay, and this whole process is also, there's a different term called mRNA splicing. So splicing of mRNA means removing a part, okay, and then removing some part and join together. Okay, so of course, one obvious question here, I don't know if you would come across your mind, is why the hell do we do that? Why not just, why not just have this sequence encoded in the DNA and then just transcribe this? Why do you have to, remove some part and then keep some parts? Okay. So the reason being, actually, we like to say one gene for one protein. Okay. But again, okay, as a matter of fact, it's more complicated than that. Okay. So just FYI, it is not within your scope. I just want you to know that. Okay. So actually, so for your syllabus, we say one gene for one polypeptide. But what I want you to know is that, okay, so from one gene, okay, so actually during the splicing, like removing some parts, okay, so you have 4 exons, okay, so these 4 exons, they can all be preserved. Sometimes you may remove, okay, so maybe just combine 1, 2, 3, or 2, 3, 4, or 1, 3, 4, or 1, 2, 4, which means from one mRNA, I can produce different ultimate mRNA and different proteins, which also means one gene can ultimately give rise to multiple proteins. Because this is about information, you want to think, how can information be stored very efficiently? Because we know that some proteins, there will be some parts, that have the similar shape, okay? Maybe they're very similar secondary structure. So those sequences can be used for multiple proteins, right? Okay, so having this kind of mechanism, okay, so of course, this is usually only present in eukaryotes, okay? So prokaryotes, normally they don't have this splicing thing, okay? But eukaryotes, they've evolved because we need so much more proteins than a prokaryote, a very simple organism, okay? So there are ways where you can, by meat, okay? You have a long stretch of DNA, okay? A long gene, okay? Of course, that whole thing going to be transcribed. And whatever that's transcribed, I can pick different part, join together, give you different ultimate mRNA. Okay, so this is like this is the final mRNA. So from one original mRNA, through this removal of intron joining the exons, okay, you can actually give different versions of final mRNA. Different final mRNA later is going to contribute or give rise to different proteins. Okay. So similarities of between your DNA replication and transcription, of course, they both occur in the nucleus. Okay, reading of template, like I mentioned, always in 3' to 5' direction, and of course, always they will involve the DNA to be unwound and rewind. Okay, and then your subunits are joined together by phosphodiester bond. Okay, so the differences, can you please screen the table for the differences? Okay, so first of all, what enzyme is involved in DNA replication? No, it's you're at the right. Okay, so what enzyme is involved in DNA replication?
Ligase.
No. DNA is a polymer. RNA is a polymer. An enzyme that catalyzes polymerization is called? No, enzyme that causes polymerization is polymerase.
Oh.
As simple as that. So for DNA replication, it will be DNA polymerase. RNA replicate or transcription, it is RNA polymerase. Okay. So your raw material, would be for DNA replication, DNA nucleotide, and then for transcription, RNA nucleotide. Okay. And then as you see, and for your for DNA replication, both strands are used as template, but for transcription, only one strand. Okay. That strand is which strand is it? What is the name of that strand? Antisense strand. Okay, that one strand is the antisense strand. Okay. And then, okay, so for DNA replication, the whole strand is replicated. For transcription, only one section, only antisense strand is being transcribed. Okay. And then air pair with T, G pair with C, and then in transcription, air pair with what? Air pair with no transcription. U. Okay, this is RNA. Okay, transcription, air pair with U. Okay, the rest T with A, G with C, or C with G. Okay. So the product of DNA replication is two identical DNA molecules, but then for transcription, you get one mRNA. Okay, so with DNA replication, you have two identical DNA molecules. Okay. So by that DNA molecules, we mean the double helix. Okay. And product for transcription is mRNA. Okay, so next we move on to, okay, so we have now transcribed, we have copied, we have got the information in the pen drive, okay, which is the mRNA. So now how we go from this pen drive information to a protein, okay? So first, you need to know there are 3 amino acids, okay, in the cytoplasms, okay? And of course, yesterday we have also learned about transfer RNA, okay? So then and also yesterday we said that the transfer RNA, there is, for each tRNA, there is an amino acid attached site. And what amino acid is attached here depends on the anticodon, okay? So anyway, so there's this enzyme called amino acid tRNA synthetase, okay, which would attach the amino acid to the tRNA, okay? So you, hold on. Okay, and this process require ATP as well. Okay, so so this diagram summarizes that. Okay, so you need an ATP which would actually help to transfer this amino acid to the tRNA. Okay, so ATP is involved. Okay, so there's a specific enzyme. When we say specific, if we have 20 amino acid, okay, each amino acid we have a different enzyme. Okay, and of course this enzyme must somehow also recognize this anticodon. Okay, so therefore, for a particular tRNA, it will always connected to, or the particular anticodon will always be matched with a particular amino acid. Okay, so that is something very, very important because, like I said, we want to say, for example, okay, let's now go back to this table. Okay, so if your mRNA is UUU, okay, if mRNA is UUU, the anticodon has to be AAA. Am I right? Okay. So you need an enzyme which when it reads AAA, it will only attach. So there is a T. So let's say if you have a tRNA, okay, which reads AAA here, then it will only attach phenylalanine, okay? Or if you have another anticodon, okay, you have another tRNA, okay? If you have another tRNA which reads, maybe you can tell me, okay? So if you have a tRNA which reads GGA, can you please tell me what amino acid is attached? If you have an anticodon, if you have a tRNA, the anticodon is GGA, can you please tell me which amino acid will be attached?
Yeah.
Sorry? You have to look at the table.
GGA.
Pro, okay? No. Like I say, anticodon is complementary. So this is mRNA, okay? This is mRNA. Whatever that we have, the table here is codon. This GGA is the anticodon. So you need to look for, so GGA would be complementary to a CCU, okay? So, whenever this enzyme, if it got a tRNA that has a GGA, it will attach proline, okay? So it is by this kind of complementary base pairing, you can be sure this particular set of mRNA, first, it will only associate with a particular tRNA, and also we have another set of enzyme that for particular anticodon, it will only attach particular amino acid. So therefore, you can ensure that ultimately this sequence will always lead to proline. Okay, so when you have this mRNA, you will only associate with this tRNA, and there's an enzyme which makes sure that when it gets this tRNA, it will only attach proline. So therefore, you ensure CCU give you a proline at that particular position. Okay, so that's what happened. Okay, so that at the charging, okay, sometimes this process is called amino acid activation, or we say we charge the tRNA with an amino acid. Okay, so we charge a tRNA with an amino acid. Okay, so after that, okay, so then it's a translation. Okay, so translation is where this mRNA is going. Okay, so the final mRNA, after all the splicing, taking away introns, joining together with the exons, okay, then that final piece of final mRNA is going to leave the nucleus through the nuclear pore and go to the cytoplasm or go to the rough ER where they are bound to ribosomes. Okay, so ribosomes are the protein translation machinery, which means that they are the cellular organelle which makes protein. Okay, so again, so now they bind to the 5' end. Your ribosomes read from a 5' to 3' direction, which is opposite in direction to your RNA and DNA polymerase. Okay, RNA and DNA polymerase read in 3' to 5' direction. Your ribosomes read in 5' to 3' direction. Okay, so your ribosomes will cover two codon at a time. Okay, so this diagram shows, okay, it will cover two nucleotides or two codons at a time. Okay, so which means, okay, look at this diagram. Okay, so when your ribosome, if it covers two codons, okay, then it will have two tRNA associated. Okay, are you with me? Okay, so when you're covering two codon, then there will be two tRNA bound to here. Okay, so each of these tRNA will bring two amino acids in. Okay, so then RNA polymerase is going to catalyze the formation of peptide bond between the two amino acids, and then it will move to the next one. Okay, and also there are two. So the two sites where the tRNA bound, they have a different name. They are called the P site and the A site. Okay, so in the ribosome, we say there's a P site and an A site. Okay, P, I think you can just, I don't know, whatever it means. Okay, so it's the first site. Okay, A is the second. So it will always can move along. Okay, so at any time, there'll be one tRNA occupying one P site. Okay, another tRNA occupying A site. Okay, so once peptide bond is formed, you move to the next one. So the one that was A site will move to the P site. Okay, so I'll just repeat this process. Okay, I mean, not to say repeat. Okay, so here, what happens? Okay, the first three, like I mentioned, always they have the same start codon. Okay, there is only one codon for starting. Okay, it will always be AUG, and it will always bring methionine in the table that we have just now. Okay, AUG is for methionine. Okay, so then, so they're positioned at the P side of the large subunit. Okay, for elongation, then the second will come in. Okay, with the next three exposed, okay, as in they are not bound by the ribosomes, okay, and then the second tRNA will come in, okay, so peptide bond is formed. After that, this ribosome, you see, it will move. Okay, so the first one carrying methionine, so this is done, so this tRNA will be released, and then just now this tyrosine, which was at A site, now it moved to P site. So then the A site become available, another tRNA bringing another amino acid can come in, okay, and then this process is going to keep repeating until, okay, arriving at the stop codon. Okay, so basically it just keep, once peptide bond is formed, the first tRNA at the P site will be released, ribosome move. So then now the one that was the tRNA at the A side, we move to P side, A side become available, something else can come in, and that whole process repeat. So then you will have a long chain of amino acid or polypeptide form, okay, until arriving at stop codon, then this process stops, that whole polypeptide will be released, okay? So that's the whole process of protein translation, okay? So if we look at how these are the locality of this process, okay? So inside the nucleus, transcription occur, okay? mRNA is made, mRNA is spliced, which means remove intron, join together exon, and then they're going to leave, come to the cytoplasm or go to the rough ER, okay? So of course, rough ER is not shown here, okay? So that proteins will be synthesized there, okay? And also just FYI, okay, so sometimes actually when you have one mRNA, okay, to increase efficiency, you can have multiple ribosomes bind to the same mRNA. So they will just all move one bind and then move along, and then the second can bind, move along, third can bind, fourth can bind, okay? So this increased efficiency of protein synthesis, okay? We have the same template, we have the same pen drive, okay? Everyone can use, okay? This pen drive can be plugged into multiple printers at the same time, okay? You can consider your ribosomes as a 3D printer, okay? So multiple 3D printer can receive the information from the mRNA. So at the same time, you can make multiple copies of a proteins, okay? So just comparing your transcription versus translation, okay? So in terms of location, maybe you can tell, okay? So in terms of location, transcription occurs in nucleus, translation occurs in cytoplasm, okay? So the product is RNA for transcription. It can be mRNA, tRNA, or RNA. For translation, it's always protein. And enzyme involved for transcription, what is the enzyme? RNA polymerase, okay? And then for your protein, okay, so the translation is peptidyltransferase, okay? Peptidyltransferase is actually kind of a part of the ribosome. It is the one that forms a peptide bond, peptidyltransferase, okay? The bond form, phosphodiester in the case of transcription. What bond do you form in translation? You make protein. What bonds do you have in protein? Between amino acids, what bond is that? Peptide bond, okay? It's peptide bond. Okay, and then the next one. The basic unit of transcription, okay, is RNA nucleotide. For translation, transcription is RNA nucleotide, AUCG, basically the raw material, okay? So translation, there'll be amino acid, okay? Your template, okay? So transcription, the template will be antisense strand of DNA. For translation is mRNA, okay? Direction of reading the template, okay? So like I mentioned just now, transcription or RNA polymerase, DNA polymerase always read in 3' to 5' direction, but somehow, you know, ribosomes just work in a different direction, 5' to 3'. Hold on. Okay, so this is just a summary of this whole 3, okay? Replication to transcription to translation. Okay, it's kind of just a summary of whatever that we have covered before. So I won't go through this table. Okay, we'll take a short break here, then we continue. 1 minute. We are going to look at this video as a summary of I Hold on now.
Here is a cell, the basic unit
Okay,
of living tissue.
okay. Okay, so
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus.
so you see, actually,
The DNA code
if you
determines the
look,
order.
the first one was an AUG. So always start with AUG. I don't know if you saw that just now. Okay, it always start with AUG for mRNA. Okay, so of course, this is the anti-strand. So on the anti-strand, it will read TAC.
Code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA.
Okay, so she didn't mention, okay, those parts removed are called. What are the parts removed called? Go to your notes and find out what are the name. Okay, what do we call the parts of mRNA that is removed? After transcription. After transcription. Or maybe at the end of transcription. Oh. Okay, under the post-transcriptional modification, just now you draw like the blocker, the exons, introns. Okay, so the part that are removed are called neutrons. Okay, so okay, so and then the part joined together are called exons.
Then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a...
So you do see that every time only two tRNA would be on the ribosomes. Okay, so and then once the peptide bond is formed, and then the tRNA is free.
growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay, so that would be the whole process of protein translation. Okay, so kind of just summarize what we have gone through just now. Okay, so next we'll go to the next part of it. Okay, so next one has to do with the mutation. Excuse me. Excuse me, sorry. Okay, so next mutation. Okay, so I think very often now in pop culture, you always see the term mutation. Okay, so like in X-Men, Marvel, whatever. Okay, so we always see that. Okay, so they talk about gene mutation. Okay, so as we know, ultimately gene defines or maybe carries information or actually carry information for protein. Okay, so if there is a change in your DNA sequence, it basically change the protein amino acid sequence in the protein. Okay. Once the amino acid sequence is changed, all your primary, secondary, tertiary, quaternary structure can be affected. When there's a change in structure or shape, then there's a change in function. Okay, so mutation is not always something very welcome, as we know. Okay, sometimes it can cause disease. For example, cancer is a disease due to mutation in genes. Okay, so it depends on the protein that is mutated. Okay, depends on its function. Sometimes it can have better or sometimes has a worse effect. Okay, of course we are mostly aware of the bad mutation. Okay, so the good mutation are not always discovered, to be honest. Okay, unless you go and study. Okay, but whenever there is a bad mutation coming up, then there are more regularly detected, because when something wrong happens, we want to find out why, and then we realize, oh, it is because of mutation. Okay, so therefore when this so in this section we're going to see a different kind of mutation. Okay, so but fundamentally mutation refers to change in the sequence of bases or nucleotide, okay, in a particular gene, and that will give you a new allele. Okay, so allele refers to alternative version of a gene. Okay, so like you have come across this term in high school, definitely. Okay, so when we learn about genetics, you have tall plant and short plants. Okay, so we have a gene, or maybe we call it an allele for tallness and allele for shortness. Okay, basically they will be, okay, so both genes are related to your height. Okay, but they will code for slightly different proteins that gives you different what we call phenotype. Or let's say we talk about the other day, we talk about the you know, this color pigment in our, okay, in our hair, okay, or in our skin. Okay, so it's due to a protein. Okay, so there are two versions of this protein. Okay, maybe so because of mutation, this protein become colorless, as in not to say colors, it no longer absorbs light, it just reflects all light, then it appears white. Okay, before this it may absorb light, okay, because of its structure. Okay, then it is dark in color. Okay, so so that's what we call a different allele. Okay, so different version of the same gene. Okay, so new allele always arise from mutation. Okay, so of course this mutation can either be inherited or not. Okay, so for example, so there are certain mutation which, okay, so it really depends on where the mutation occurs. If the mutation occurs in the gamete cell, okay, then which means the reproductive cell, then they'll be passed on. Okay, so let's say if it's in the sperm cells or ovum cell, okay, so that mutation will pass on to the next generation. Okay, but if it's in somatic cell, you know, for example, like maybe there's a mutation in the skin cell, okay, so that wouldn't be passed to the offspring. Okay, so those cells which are not gamete cells, okay, not reproductive cells, we call them the somatic cells. Okay, so that's why here it says somatic mutation. Okay, so any cells other than reproductive cells and those which can undergo mitosis but not meiosis, okay, are called somatic cells. Okay, So here we learn different kind of mutation. The first is substitution, okay? So in substitution, you basically just substitute, just change one base. Yeah, you can take this down. Yeah, I want you to just also take those notes down as well. Okay, so here, like we say, so if you look at this table, okay, so we know that if you change a nucleotide, okay, sometimes a change in a single nucleotide, okay, can cause severe problem. Okay, so let's say, for example, if you have, okay, originally, if it is, let me see, okay, so let's say if you have an AUU, okay, and then maybe this A become a U, then it becomes a phenylalanine, okay? But there I say it may change amino acid sequence. Why? Because it really depends on what position where the mutation take place. If let's say it takes place at the third position, okay, very often it may not cause a change in amino acid because it depends. Okay, so if U change to C, there is no mutation, as in there is no change in amino acid because both of these have this code for the same amino acids. But if you change to A, oh, then you change a different amino acids. Okay, so depends here. For example, like CCU, you know, if whether if it's happening at the third position, it wouldn't change the protein into another amino acids. But typically, especially mutation on the first and second position of the codon, very likely leads to a mutate change in amino acid. Okay, but yeah, sometimes it is silent, as in like when we say some mutation is silent, that means it doesn't cause any change. Okay, so doesn't this case, so yeah, so this, okay, by the way, this kind of mutation, we call them the silent mutation. They are silent because they do not cause change in amino acid. Okay, no change in amino acid. So when it doesn't cause a change in amino acid, we call that a silent mutation. Okay, so you will not notice it. Okay. And then there is this deletion, okay? So deletions and addition simply basically you remove some part of amino acid or you just add additional amino acid, okay? So addition is also called insertion. So for both these kind of mutation, they can lead to what we call frame shift, okay? I'll talk to you about frame shift in a minute. Okay, we'll talk about frame shift later. Okay, and oh, maybe I'll just come to you. Okay, so let's look at here. Okay, So let's say if this is some of my, okay, so let's say because it's triplet of codes, okay, so maybe this is the wild type, as in like the healthy one, okay. So original one, you know, it reads dog, cat, cow, pig, B. Okay, if suddenly the C mutated to B, okay, there's only change in one amino acid, so this is substitution. Okay, so for frame shift, okay, we call it deletion. Okay, so let's say it really depends on how the deletion occurs. So let's say here, okay, here from here to here, you delete two base. Okay, so originally this would be correctly read as dog and cat, but after that, from the third position onwards, it no longer makes sense. It doesn't even read like an animal name. Okay, so frame shift basically causes your change in reading frame. So instead of reading, so basically it just shift. Long story short, once frame shift happens, everything follows changes. All the amino acid that follow can change. Okay, will change in their identity. Okay, so the whole sequence will change. Okay, so any sequence following frame shift mutation will be read differently, and then you will have a totally different proteins, and very often non-functional. Okay, so same thing with addition. You add two more, okay, but sometimes let's say, okay, if let's say here if the deletion involves a multiple of 3, okay, let's say it removes I don't know, here it removes ATW, okay, ATW, okay, or maybe no. Okay, let's say for this one, okay, it dot T. Okay, so let's say, oh, hold on. Where does this come from? Let me see. Oh, this is not a good example, but I'll just change this a little bit. Hold on. Let's say change this to. Okay, let's say I remove this TCO3. Okay, so here you change, but for the following, it is still normal because this frame shift, okay, only so because this deletion involves a multiple of 3 nucleotide. Okay, so in that case, okay, so whatever that follows may not change. Okay, or let's say if you have an maybe you just totally lose CAT, so that's why you see you simply lose one amino acid. There is no frame shift, which means the reading frame for the subsequent nucleotide is still the same. I still get the P, I still get the B, but I don't change anything. Okay, so if the addition or deletion happens in a multiple of 3, it doesn't cause like a frame shift such that, you know, everything else changes beyond. Okay, so here, okay, so frame shift is ribosomes begin to read wrong triplets from the point of addition or deletion. Okay, so of course, very often this can cause formation of a non-functional protein. Okay, or sometimes it may cause an early arrival of a stop codon, as in that amino acid just stop reading. Okay, so it will create a new an early stop codon. So instead of maybe a protein of 1000 amino acids, you get a protein of, I don't know, 600 amino acids, let's say. Okay, so just you can take this.
So for drawing part, you will send to the group, is it?
Okay, I'll send to the group later. Okay, maybe you just take the text, it's fine. Okay. Maybe the rest I'll send it to you later. Okay, so I'll just move on. Okay. So next is a sickle cell anemia. Okay, just one example, so that will finish this chapter. Okay, so sickle cell anemia, okay, is caused by a mutation in the gene which codes for beta globin. Okay, so hemoglobin, as we learned before, is a quaternary structure. Okay, so it has 2 polypeptides. So one of the polypeptide is called beta globin. So basically, what happened is that, you know, there is a change in a nucleotide sequence, and that leads to a change in a polar amino acids to a non-polar. Okay, so basically, you know, hold on, your okay, so this is a glutamic acid amino acid is changed to valine. Okay, so you are not expected to remember the name or know the name of amino acid, but when you see one, you need to recognize, oh, when you see this is an amino acid. Okay, this is also an amino acid. Okay. Okay. So long story short, okay, because of the change, a single amino acid, okay, you have what we call a sickle beta globin. Okay. Long story short, this thing, it becomes because of that change, okay, simply, flipping a polar to a non-polar, it affects the three-dimensional shape of the protein. It can no longer bind oxygen very efficiently. So patients carrying this mutation, okay, so of course they would have a deprived ability to carry oxygen, especially if they are more homozygous, as in if this mutation occurs in both genes, or maybe if they inherited from one from dad and one from mom, let's say both mom and dad may be a carrier for that particular allele, okay, and if that kid inherits both copies, okay, then if he has two copies of this, so these people tend to be very sick, okay, so you have one copy of healthy one copy of a sick one, it may still be okay. So normally they may look normal, but if only when they exercise very strenuously, then they may have some anemic symptom. Anemic, you know what is anemia, right? Okay, anemic basically refers to people lacking it's Chinese we call 贫血, basically lack of blood, but actually it's when you have lack of that, okay, so you have a reduced ability to carry oxygen, okay, so these people, they cannot have very strenuous physical activity, okay. So this is, you see, this is a simple substitution mutation. Only one nucleotide change causes severe problem. And also somehow because of the change in the nature of the hemoglobin, it kind of it also affects the cell membrane, maybe because it become more non-polar, it would associate with the plasma membrane, and then the cell normally appear in this kind of shape, okay. So sickle cell, this is a sickle shape, okay. So for patients with this disease case, so they no longer have the biconcave shape, okay. So the red blood cell have a sickle shape, okay. So that's why it's called sickle cell anemia. So here you see how the change in a single amino acid, a mutation, can cause severe problem. Okay, so with that, we are done with this chapter. Okay, so I'll screenshot this and then send it to you. Okay. Okay, with